everyone. Thank you for joining. Welcome back to the next webinar in our PLANTAE Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by PLANTAE, the open online community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who may not have joined ASPB yet, you can join today and use the discount code PRESENTS10 to receive a 10% discount on your registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. We started this series last year. In 2020, we held 25 PLANTAE Presents events with 59 speakers in a wide range of topics. If you haven't already done so, do check out our archive to watch out all the previous talks in this series. You can find information about each event on our plantae.org blog and subscribe to our PLANTAE YouTube channel to get notifications when new recordings are posted. We have 6 more upcoming events listed on our website for 2021 and more to come. We encourage you to attend them all. Next week, we will have a second panel discussion about specialized metabolites and their roles specifically in defense. If you need to leave this webinar early, know that a recording of the webinar will be made available within a few days. Today, we have 4 great panelists joining us. Each of their talks will be 7 minutes, followed by 5 minutes for questions. At the end of today's session, all of our panelists will join together for an extended Q&A. As you listen to each talk, add your questions to the Zoom and question and answer Q&A section. Questions that are specifically for our panelists can be shared within the Q&A to make them easier for our moderator to navigate. You'll also be able to upvote and comment on the questions there. You'll notice that there's also a chat area open. This section is for you to introduce yourself, say where you're from, and connect with your colleagues. All right, so without further ado, I'm going to hand it over to our wonderful moderator and PLANTAE Presents steering committee member, Gitanjali Yadav. Gita is a lecturer in the Department of Plant Scientists at the University of Cambridge, and she is a scientist at the National Institute of Plant Genome Research in New Delhi. She will be introducing our speakers and collecting your questions. Thank you so much, Gita.
Thank you, Katie, and welcome everybody to the first part of PLANTAE Presents panel on specialized metabolites today. Plants are amazing chemists, and they've evolved a fantastic array of specialized metabolites with diverse functions. Some have role in development, others in protection, defense, attraction of pollinators, and some have also functions that we haven't discovered yet. So today we are going to look at a few of these fascinating small molecules with 4 of our panelists, and the rest of it would be on Feb 3. Our first panelist, whose work you're going to be listening to just now, is Gregoire Loupit. Greg will be talking to you about understanding graft union formation and graft incompatibility in the grapevine. He's currently a PhD student in France, working on understanding, of course, graft union, and his subject is managed by Sarah Cookson and Josep Valls in the Institute of Vine and Wine Sciences in France. One part of his thesis was to identify marker metabolites of grafting success, especially by analyzing stilbenes at the graft interface. And this is what you're going to be listening to now. Katie, can we have the slides, please?
Hi everyone, my name is Gregoire Loupit, and I'm going to talk about my PhD subject, which is understanding graft information and graft incompatibility in grapevine. The project was done at the University of Bordeaux in One and Vine Science Institute. So, as you know, grafting has been used for millennia for vegetative protection or to provide resistance to biotic or abiotic stresses. And for grapevine, we use omega grafting, as you see on this little video. And grafting is also used on woody spices like citrus, apple tree, pear, but also on herbaceous spices like Arabidopsis or tomato. In grapevine, rootstock was to provide resistance to phylloxera, a pest that appeared at the end of 19th century. So during grafting, we can describe three stages of development that involve many different gene families. In growth and development for callus formation, and in secondary metabolite for defense mechanisms. So the first step is the development of necrotic lines at the graft interface in response to wounding due to an oxidative stress and the closure of vessels. We have also some compounds such as lignin can act as physical and antimicrobial barrier. Next, there is a proliferation of parenchymal cells for callus formation and for mechanical structure. And finally, there is the formation of vascular vessel, which allows a connection between the two genotypes. However, there are some incompatibilities after grafting, and sometimes symptoms can appear several years after planting. These incompatibilities are associated with more necrosis and poor vascular connection, as you can see on picture on your right side. We know that there is many incompatibility causes, like genetic distance between the two genotypes, but also quality of the graft, climatic condition during the growth, or the presence of viruses can be also other factors of incompatibilities. In addition, we believe that difference in metabolisms between the two genotypes could also explain some incompatibilities. Today, it's very difficult to have visual indicators to know if a graft will survive or not, so it will be very interesting to find biochemical markers of graft incompatibility in order to help wine nursery industry and, for example, selection of rootstock. So to do that, we analyze 9 enterograft and 6 homograft control at 33 days after grafting from the French Institute of Wine and Vine. So we had 3 science like Marrow, Negret, Uniblanc, and 3 different rootstock, SO4, RSB1, and 140 Regery. And all these cultivar and species with different degree of compatibility, as you see, for example, Marrow and to SO4 at a high percentage of success rate compared to Uniblanc and to SO1. This percentage was calculated at the end of the year in December using the DIM test to test the solidity of each grafted plant, like on the video, as we can see. And to find markers, we studied and quantified some primary and secondary metabolites. We quantified different amino acids, starch, glucose, fructose, and different families of phenolic compounds, in particular stilbenes. All of these metabolites were analyzed from 3 parts of the plant, which was at the graft interface, scion, and rootstock. So stilbenes are known for their antioxidant and antifungal properties. The main compound was resveratrol, but other monomers, dimers, trimers, or even tetramers can be found. In analogy unit, a new method of quantification by HPLC coupled with a triple quadruple has been developed to quantify more than 50 compounds in one single run. So our first results show difference in interface profiles compared to scion and rootstock, as you can see on the principal component analysis. At the graft interface, there is an accumulation of proteins, several amino acids like alanine or serine, soluble sugar, especially fructose, and stilbenes like alpha-viniferin or parnellocysine, for example. So to identify markers, we made some correlation matrix, especially regarding homograft, a high correlation was found between several metabolites in the cyan and the percentage of grafting success, as you can see with red arrow. However, no strong correlation was found in other tissue, and no correlation was found in heterograft combination. For example, here, transresveratrol and pisathanol increased in compatible combination, and alpha-vinylfurin was more concentrated in incompatible combination. So we think that this compound can be a very good marker of grafting success only 1 month after grafting in homograft combination. In order to confirm these first observed results, a generalized linear model was made to estimate the correlation of a prediction model with our data. As we can see, it is the data with homograft and at the level of the cyan tissue that allow to the best correlation with the model. So to conclude, the 3 stilbenes described will need to be analyzed in more combination to check their repeatability, and other further study must be done to find markers in heterograft combination. If you know more about markers of successful grafting formation, we can check our recent review. And I want to just thank people who work with me, and thank you for your time.
Greg, that was a very nice presentation. So very interesting that you have shown a correlation and such a high correlation between certain chalcone synthase products, I would say. You have shown in your slides that resveratrol and pisathanol are both good markers for grafting success. Do you think it is possible that the presence of these compounds are not the result, but the cause of the good graft? And in that case, would it be possible that supplying these compounds at the time of performing your graft could make even your less compatible or less successful grafts that you tested today in your presentation, could it make them work also? What's your take on that?
Hi, nice to meet you, and very happy to participate to this panel. And you write very good question. I don't know if these compounds are the result or the causes of grafting success, but this is why next step of my PhD was to study this question. So I want to test the addition of these compounds, such as you say, resveratrol or pisathanol, during grafting, and try to increase the percentage of grafting success. But also I want to try to test the addition of maybe some elicitor of stilbene, and try to also improve grafting success. And another question that's also interesting, that I don't know if this compound can improve grafting success, but to answer with this of the question, we also want to study a kinetic of accumulation of polyphenol at the graft interface during the first month, to see differences of accumulation between different genotypes, and maybe understand why some combination are compatible or not.
Can I ask you, there's this of the grafting of monocots. That's usually considered the holy grail. And seriously, in case of serial crop transformation, at least, I think it's still a question that for years and years has kept people busy, and lots of researchers have tried to try and get a serial crop transformation done with the help of grafting. What are your views on generation of such grafts without regard to cultivar or genotype? And why do you think it's been so difficult to achieve that holy grail or efficient methods for monocot grafting?
You're right. I'm not a specialist about cultivar than grapevine, but I think monocot, it's a special case of plant family, because they don't have vascular cambium. And cambium, it's essential to form a callus for graft formation and to have the connection between the two genotypes. And I also read one time that one possibility of that one possibility is that during evolution, monocots have lost the ability to graft. In viticulture, grafting is for now essential because of the phylloxera. You can improve so quickly yield, vigor, many of the factors, and for example, you can choose one rootstock which is adapted to a type of soil, for example, and keep the cultivars that you want.
Yes, I completely agree with you there. In fact, my last question, let's see the time. I think I have time for one more question to you, especially with regard to my own research. And therefore, I think, so for lots of years, for a long time, we've been trying to quantify relationships in the chemistry, in chemical compounds. And a lot of work or published work in this area is basically about comparing chemicals by using the Bray-Curtis index or looking at the sizes or the names of the chemicals. You have in your work vectorized chemical features because you showed us some nice PCAs with your chemicals. And so how did you, in your correlation plots and metabolite PCAs that you've got, how did you featurize the chemistry? And would you have any ideas or tricks or tips for our listeners on quantifying chemical compounds or quantifying phytochemistry?
Yeah, just in our study, we don't use features because we use real correlation in your extract because we use integrated metabolic approach. And in fact, with this method, we use commercial standards, and some of them also we are purified in the lab. And so with this method, we have real concentration in milligram per gram of fresh weight. And so to construct PCA and also correlation plot, we use real correlation. But it's good to talk about features because it's very interesting to use, for example, an untargeted metabolic approach. And with this method, you can have full scan of your extract and compare chromatogram between compatible combinations, for example. And if I can give one tip, suggestion, maybe for create a PCA, don't try to put many variables and different data in your PCA. For example, if you want to separate cultivar and also different years and different condition, etc., it will be complicated to interpret. Maybe.
I see. Okay. Thank you so much. Thank you so much, Greg. With that, I'd like you to meet our next panelist of the day, Dr. Benjamin Lichman. He's currently working at the York University in UK. Benjamin received his master's in natural sciences from the University of Cambridge, and then he did his PhD in biochemistry from the University College London. His doctoral research, under the guidance of Professors John Ward and Helen Hayes, focused on, that's right, okay, Katie has already put his video. Katie, please carry on that. Yeah, let's begin the video, please.
Sorry about that.
Hello, my name is Dr. Benjamin Lichman, and I'm a research fellow and lecturer in the University of York in the UK, and I investigate plant specialized metabolism. My group takes a very broad view of plant specialized metabolism, investigating the molecular mechanisms, including discovering biosynthetic pathways, looking at enzyme mechanisms. We also look at the evolutionary origins of plant specialized metabolism, such as how different enzymes evolve, how gene clusters are formed, and how different compounds are formed across different species. And finally, we also take a look at the practical applications of plant specialized metabolism, including different ways to produce high-value compounds for use in industry and pharmaceuticals. Today, I'm going to talk to you about a project that I started whilst I was a postdoc, working with Professor Sarah O'Connor, who's currently at the Max Planck Institute for Chemical Ecology in Jena. And it is the work that we've been doing on catnip. So the title of the talk is The Molecular and Evolutionary Origins of Nepetalactone Biosynthesis in Catnip. So what is catnip? Well, catnip, also known as Nepeta is a plant from the mint family, and it's famous or infamous for its effects on cats. And you can have a look at the internet and see many videos of cats interacting with catnip. It sends them into some sort of state of euphoria, and they roll around in it and generally seem to enjoy it quite a lot. What is the reason, what's the origin of this effect? Well, the origin of this effect is a chemical called nepetalactone. And these are structures of 3 different isomers of nepetalactone. This large compound here is called cis-trans nepetalactone, and the cis-trans nomenclature refers to the different stereochemistry of the compound. And so catnip, or Nepeta and related species Nepeta mucini, produce 3 different isomers of nepetalactone. You see cis-trans here, cis-cis nepetalactone, and trans-cis nepetalactone. What's notable is that different individuals and even different parts of the plant seem to produce different ratios of these isomers, as you can see a GCMS trace here showing different isomers from different individuals. And we think that the real reason why the plant produces these compounds is to interact with insects, probably repelling them, possibly attracting them, generally to prevent herbivory. But there's still some work that needs to be done in that area. So these compounds are volatile monoterpene iridoids, and they derive from geranyl pyrophosphate and geraniol, but rather than using a typical monoterpene synthase type pathway, they're this non-canonical pathway, which is kind of characterized by the presence of this enzyme called iridoid synthase that forms the iridoid bicyclic structure. So we've used a number of different enzyme discovery methods over the last 3 or 4 years, attempting to discover enzymes that are involved in the nepetalactone biosynthesis. For example, we've used homology to find iridoid synthases. We also use proteomics, so isolating trichomes from the Nepeta leaves and basically finding which enzymes are most abundant in these sort of chemical factories that are the trichomes. And we've also been more recently using genomics and looking for gene clusters and finding enzymes associated with biosynthesis in gene clusters. So what have we found? Well, these are the 3 isomers. And what we found is that these 3 isomers in Nepeta all kind of start with this origin in 8-oxogeraniol, and this compound is reduced by iridoid synthase, and iridoid synthase actually produces this unstable intermediate. We then have these cyclase enzymes that cause divergent stereoselective cyclization of this intermediate into these 3 different nepetalactol isomers. And then we have dehydrogenases that take these nepetalactol isomers and oxidize them into the final nepetalactone products. We can replicate this pathway in vitro, and you can see here different combinations of different enzymes that cause formation of these different isomers. We also see it in the plants themselves. If we take different plants that produce these different isomers, we see corresponding changes to gene expression. For example, the trans-cis producing plant has a high expression of NEPS4 and NEPS1, which fits with this pathway. So now we have, what are the enzymes that we're showing here? So in particular, we found this subfamily of short-chain dehydrogenases that we call the NEPS enzymes. And some of these are typical dehydrogenases, but we also have some of these that seem to catalyze this reaction that we call a 4+2 reaction. This 4+2 reaction is also known as the Diels-Alder reaction and is quite an unusual enzyme reaction, and especially unusual to be catalyzed by dehydrogenase enzymes. We have a crystal structure of one of these cyclases, and currently in the O'Connor lab, they're working on trying to understand the mechanism of this enzyme. But I'd like to kind of highlight how it kind of shows, this work shows the plasticity of enzymes, and this plasticity of enzymes, especially within plant specialized metabolism pathways. So we think that a dehydrogenase essentially evolved to become a cyclase, and this really shows how new enzyme activities can evolve from seemingly unrelated enzymes. So I mentioned evolution there, and we are quite interested in evolution, and especially in the interactions between enzyme evolution and genome evolution. So we found these gene clusters on the genome of Nepeta cateria and Nepeta mucini, and we basically find these NEPS enzymes close by to iridose synthase enzymes. So we decided to work out how these iridose synthases came to be in this genetic, through this genomic locus. And we did this by taking the enzymes, looking at all paralogs that are present within Nepeta species and closely related species, and testing them in vitro for different activities. We also resurrected some of the hypothesized ancestors of these enzymes as well and tested their activities. And essentially, these three iridose synthases here are the ones that we find in the cluster, but these paralogs are not found in the cluster. And by kind of combining all this information, we're able to build a picture of Nepeta-lactone pathway evolution and also the genome evolution as well. So what we propose is that we have two interesting loci. One we call the progesterone beta reductase locus, and one we call the NEPS locus. And essentially, in this progesterone beta reductase locus, we have an ancestral enzyme that can do some of the iridose synthase activities, got a relatively low activity. But after duplication and a period of positive selection and neofunctionalization, it evolved into an iridose synthase. And it was actually only after this point that it moved into the gene cluster. And so at the original locus, that it then underwent pseudogenization. And so this is an interesting example where we see that enzyme evolution appeared to evolve before the gene cluster evolved. And this is something that my group are currently investigating further, how a gene cluster has formed within the Nepeta. So thank you very much for listening. I just want to thank the O'Connor Lab and Robin Boyle, who's was doing a lot of the genomics work, and the Mint Evolutionary Genomics Consortium, who have contributed massively to this work. Thank you very much for listening.
Let's get to know you first, Benji. So what was the first key moment in your life that turned you on to plants? And what is it about your current project that connects you to that moment?
Okay, so to be honest, my background is not really within plant biology. So at university, I didn't. So I didn't study plant biology. Instead, I was doing organic chemistry and enzymology. And so it was actually during my PhD when I started working on an enzyme from the morphine biosynthesis pathway that I got interested in plant specialized metabolism and then plant biology from that point. So although I was kind of focused on these enzymes, I kind of saw how complex and exciting and interesting plant specialized metabolites can be, and then linking this with the genome and linking it with kind of the adaptation of the plant and the way that these chemicals interact in an ecological setting. I think that kind of has made, in the last few years, I've got much more excited about plants than I was beforehand. And there are a few, there's a paper especially that I read that kind of really triggered a lot of this interest, which was a paper it was by Edgar et al., where they showed the Brassicaceae and how the evolution of these glucosinolates kind of matched to what butterflies were kind of eating them. And this sort of link between interesting chemistry, interesting enzymes, and ecology is something that I'm really interested in.
That's lovely. It makes so much sense when you start to ask people questions about the background of the research they're doing connected to their own lives. I think that gets you connected to the person you're listening to or speaking to. Me, myself, I've had a huge diverse educational background, but coming back to plants all the way. So I too did, stereochemistry, biomedical research, immunology, all over the world, and then back to plants altogether. And that's a story for another PLANTAE panel, possibly. So my second question to you is, you're very well aware that last week there was a new research published in the journal Science Advances that suggested that cats not only use catnip for a high, just the way you talked to us about it, but they may also use it as protection against mosquitoes. In fact, feline attraction to iridoids has puzzled, as you told us, researchers for many, many years. And we are still unsure why these party drug-like effects are not observed in other animals like dogs or mice. I'd like some of your views on what your take is on this, especially because the authors in this paper mentioned that, in fact, they argued that the iridoids have aided behavioral evolution in cats. Would you agree? And what's your comment on that?
Okay, so yes, the paper that was released last week was really interesting and sort of very, very exciting for the catnip field as a whole. I'll kind of approach there's many parts to the question, but I guess I'll start with the question about, why aren't dogs excited by catnip? And I think it just comes down to the fact that, dogs and cats have different physiology, they have different behaviors, they have different evolutionary history. So I don't think there's any particular expectation that these different animals would kind of have, the same response to different chemicals. In terms of the sort of evolutionary aspects and the cats, yes. So we kind of knew, but before the paper came out, we were aware that volatile iridoids had insect repellent properties. So this is probably what's going on within, the evolutionary origin for the reason why the plants make these compounds is probably to repel insect herbivores. So we knew that was a property, and there have been a number of studies showing mosquito repellency. We also obviously knew that cats were attracted to it and kind of, gained this sort of euphoric response. But really it was this paper that put the two together. They showed that when cats rub themselves on catnip or silvervine, in the case of that paper, the compound is transferred onto the cat's fur, and then that causes the cat to kind of repel mosquitoes. So it was really just kind of putting these things together that the paper did really well. I'll be honest and say that until this paper came out, I was pretty convinced that it was a coincidence. These volatile iridoids just so happened to attach to a particular receptor, and it was just kind of some sort of chance or coincidence. But I think there is some argument to be made for some sort of adaptive evolution on behalf of the cats. It's possible that this behavior evolved around a time when the cat ancestor was in the presence of plants that were producing volatile iridoids. And I could imagine that there could have been over time a response that evolved within cats to attract them to the plants and to kind of gain this mosquito repellency. And what's really exciting is that the timing just about matches up as well. So I think panthers are also attracted to catnip, and so like tigers and lions. And I think that the ancestor of these species was probably around 10 to 20 million years ago. And also in kind of like Western Asia or sort of Iran area. And this is around the time that we think that catnip evolved as well. So there's something maybe going on there that's worth further investigation.
Very interesting, and a lot of food for thought. So my last question to you just now, and of course we're going to be taking audience questions at the end. So if you have questions for Benji, please put them down in the chat box. So my question to you is the ancient genie in a bottle question. If I could introduce you to an all-knowing, all-powerful genie that, with one wave of her magic wand, she could instantly remove one bothersome bottleneck, one that's been preventing you from conducting your research with the ease and fluidity that you wish you could enjoy right now, what would you ask for and why?
Okay, so I think that I'll kind of introduce a different project that my lab is working on at the moment. So we're investigating some complex alkaloids. Essentially, we're just trying to do some gene discovery, finding out how these plants, what the enzymes are that form these complex chemicals. And the gene discovery aspect is certainly a bottleneck. However, I also think that that's kind of a fun part of a project. That's kind of where some of the excitement is. When you might screen through 20, 30 genes, and when you discover that gene and kind of see the activity, that's quite exciting. So although that is a bottleneck, it's not something that I would kind of wish away. Instead, I think for us at least, I think the chemical synthesis, chemical identification, chemical quantification. So when you're doing this, untargeted metabolomics, or even working on an enzyme that you hypothesize catalyzes a particular reaction with a complex substrate and a complex product. Getting hold of those compounds can be very challenging. And especially if sometimes you can isolate them from the plant. It's quite rare to be able to actually form them using chemical synthesis just because of sometimes their complexity. But if you're kind of in the middle of a pathway, you need an intermediate that is really not very highly abundant within the plant, so you can't get hold of it so easily. So I think my magical genie would be able to essentially allow us to make, all we're asking for is 10 milligrams of a chemical that we could have in a vial and use as a substrate and a chemical standard for our assays.
Thank you. Very nicely handled, that one. In fact, the gene discovery part of your wish at the beginning, which you had mentioned at the beginning, some of it might be answered by the next talk, where Igor Cesarino, professor at the Department of Botany at the University of Sao Paulo, is going to talk to us about lignin metabolism and his work on systems biology of this entire process. Igor is currently working on the molecular mechanisms underlying lignin biosynthesis, as well as regulation, and on the engineering of secondary walls to generate optimized biomass crops. Igor is also a plant physiology assistant features editor. Katie, can we have Igor's slides, please? Thank you.
So today I'm going to talk a little bit about using a systems biology approach to study lignin metabolism in plants. So lignin is a phenolic polymer deposited in the secondary cell walls of specialized cell types, where it provides three biological functions. First, lignin provides physical strength to supportive tissues. Second, lignin waterproofs water transporting cells from the xylem, allowing the transport of water and nutrients throughout the plant body. And third, lignin is a physical barrier providing protection for plants against herbivores and pathogens. Not only lignin is important for plant physiology, but it is also important in the perspective of the bioeconomy. So in the bioeconomy, the idea is to use plant biomass as a feedstock for the production of several downstream products, because plant biomass is enriched in cell walls. In turn, cell walls are enriched in cell wall polysaccharides. Those carbohydrates, structural polysaccharides, can be degraded into monomeric sugars, and these monomeric sugars are fermented for the production of biofuels, biopolymers, and a plethora of other products in the biorefineries. However, lignin is a bottleneck in this process. It hinders the efficient processing of plant biomass into downstream products. So there's an opportunity here to use biotechnology to engineer lignin metabolism in order to produce optimized plant biomass for the biorefinery. So in our lab, we focus on all aspects of lignin metabolism, from the regulation of its biosynthesis, and this regulation occurs in several levels, in the transcriptional level, post-transcriptional level, and post-translational level. Then the biosynthesis that occur from the precursor phenylalanine until the production of the lignin monomers, the so-called monolignals. These monolignals are transported from the cytosol towards the apoplast, and in the apoplast they will be radicalized before they are polymerized into the final lignin polymer. So all of these aspects are also targets for lignin bioengineering. In our lab, we focus on C4 grasses, and here I present a work that we did to develop a xylogenic culture for sugarcane as a model system to study lignification in sugarcane. So xylogenic culture is a tissue culture system in which you can induce the differentiation of tracheary elements, which are xylem cells that deposit high amounts of secondary cell walls and so also lignin. So in our lab, we first developed a suspension cell culture from sugarcane stems, and then we treated those cells with auxin, with a combination of auxin and brasinosteroid. And this trigger the differentiation of tracheary elements, and we observed two patterns of secondary cell wall deposition: the reticulate type, which you can see on your left, and the pitted type, which you can see on your right. Both of them are typical of metaxylem type of xylem cells. Then we follow these cultures for 4 weeks, measuring the lignin content, and we observed that the differentiation of tracheary elements was accompanied by increased levels of lignin. So this was triggering also lignification as expected. So then we use the systems biology approach. So we follow this culture using RNA-seq analysis, using high-throughput transcriptomics, and we cluster these genes according to their expression profile. So we observed the global changes in gene expression, and we cluster those genes according to their expression pattern along the culture. And we observed that, for example, in those two clusters here, cluster 1 and cluster 2, which shows genes that whose expression increases through time in induced samples, we observed that these genes are enriched in the phenylpropanoid pathway. They are enriched in ROS. So they are enriched in GOs that are connected to xylem differentiation and secondary cell wall deposition. Whereas the genes that show downregulation in induced cells, they are characteristic of cell division, which is expected already. By analyzing specifically lignin-related genes, we can see by checking the gray bar here in every step that lignin-related genes were massively upregulated in induced cells, including also enzymes that are involved in lignin polymerization. This is corroborating the increases in lignin levels. And finally, we use these expression data to generate co-expression networks. That we are looking here for genes with unknown function that are highly co-expressed with the lignin gene. So here in this network, you can see in the red dots here, those are non-lignin related genes. The green ones are non-transcription factors, and the blue ones are genes with unknown function. Why those genes are important? Because based on the guilt by association principle, genes that are co-expressed or genes that are expressed in the same place and in the same time, they might be involved in the same biological process, which means that those genes with unknown function that are co-expressed with lignin-related genes, they might also be involved in lignification. Those are our candidate genes now. And what we are doing now is, of course, to perform functional characterization, the misregulation of these genes in PLANTAE, in C4 grasses, and analyzing whether this misregulation leads to a lignin-related phenotype to ensure that these genes are indeed involved in lignification. To do this, we are not using sugarcane because sugarcane is a very complex plant. We are using a model plant for C4 grasses, which is Setaria viridis, which is closely related to very important bioenergy crops such as maize, sorghum, and sugarcane. With this, I would like to finish, and I thank you for your attention.
Thank you, Igor. So thanks a lot for the systems biology angle to lignin metabolism. My first question to you would be to try and get to know you a little bit better before we get to know your science. So with this project that you've been doing, which is the biggest question you feel driven to answer now based on what you've based on the insights you've gained?
This is a project that I started when, of course, I started my lab in Brazil after coming back from Belgium, where I did my postdoc. And I wanted a project that could feed my research with candidate genes for many, many years. So I was actually, the major question that I have with this project and the major question that I have for my field is how lignification occurs. So how lignin deposition occurs, because this is a very complex process. As I mentioned in one of those slides, you have the regulation. One aspect of lignin that is very interesting is that plants do not have a mechanism to degrade lignin. So all the carbon and energy investment to produce lignin cannot be taken back by the plant. So the plant cannot degrade lignin and use the carbon back. So the regulation must be very, very, very well controlled. So that's why you have, only in regulation, you have so many levels of regulation. Even pathway intermediates can regulate in a negative feedback, can regulate the enzymes involved in its own biosynthesis. And then you have like how the transport of monolignals or other monomers occurs from inside the cell towards the cell wall. We barely know how this process happens. There's only one single gene characterized in this process. Also, how lignin polymerization occurs, because you have like multigenic families, gene families, and not all of them, all of those members are involved in this process. So there are so many questions still. So that's why I wanted to have a systems biology approach, to have a list of candidate genes that now we can start selecting which aspects of the lignin metabolism that we want to address, and we can search for the best candidates for functional characterization. So the question is always how lignification occurs.
Fantastic. In fact, you've chosen just the right technique then. Use of genome-wide co-expression networks is increasingly becoming recognized as the most reliable technique for extracting transcriptional gene regulatory inferences from the RNA-seq data and so on. So my next question to you is about the co-expression network data that you generated. Was it weighted? If so, how did you generate the weights? What kind of weighting method did you use? And if not, why did you use an unweighted method? Basically, what I'm trying to understand is, do you have any recommendations for people out there listening to us and wishing to use their RNA-seq for a specific trait of interest as you have tried? So any suggestions?
Yeah, our network is weighted. We use the WGCNA methods. It's an acronym for weighted co-expression network analysis, which is much more reliable than unweighted networks, because basically unweighted networks just say that whether two genes are co-expressed or not, kind of like yes or no, whereas weighted co-expression networks gives like a value for how strong two genes or a pair of genes are co-expressed, which means how similar the expression pattern of two or a pair of genes is along your experimental design. So indeed we use this weight approach. And if I have to recommend how people should do it, I have like basically 3 recommendations. First, to use very contrasting conditions for your biological process. So in the case of lignin, of course, that we use developing tissues from non-lignified until very or highly lignified tissues. Second is to have the more time points or harvest points you have, the better. So if you have like only two comparison, it's just like here and there, and you cannot really predict a lot about the behavior of the gene along the process. So if you have a lot of time points, then you can really correlate or have stronger correlations between genes. So this is much more powerful. So the more the data points, the better. And finally, if it's possible, also to include mutants. This is also very interesting. For example, for Arabidopsis, we have basically mutants for all the genes, the biosynthetic genes in lignin. So why is it important or interesting to include mutants? Because when you have a perturbation in the pathway, you have not only a metabolic feedback, because the metabolic flow would be redirect to somewhere else, to another branch of the metabolism, but you also have a transcription of feedback. So a lot of genes, the expression of a lot of genes of the same pathway will change. And normally, genes with the same function will behave in a similar way in the mutant background. So when you have a perturbation, genes with the same function would show similar changes. So if you can include several mutants, you can also use them to analyze how genes, the expression of genes involved in a similar process might behave. So this is also an alternative to increase your predictive power, not only having contrasting tissues, but also having pathway perturbations.
Perfect. Thank you so much, Igor. That's exactly what we tell our students. So I'm part of this Cambridge Bioinformatics training facility, and we've been teaching the WGCNA for the past 2 years. A lot of our students come in wanting to just know how many replicates for the RNA-seq, and people usually think 1, 2, 3 replicates is good enough. And we end up telling them 6, minimum 6. If you're planning an RNA-seq, please talk to a bioinformatics guy first. Please make sure you have a sufficient number of replicates, and all of your points perfectly fit into this. I hope some of our students currently who are planning their transcriptome experiments do take some of these tips into account. I am going to digress from my usual course right now because we've got 3 different questions for you on the chat box. So Igor, I'm going to put to you a question asked by Javier Moreno. And the question is, are you currently trying to characterize some of those unknown genes in Brachypodium? How far are you and the biofuel industry to target some of those genes in sugarcane? Would you like to
so thank you, Javier. Javier was the person who invited me to subscribe for this event. So I thank you, Javier, for inviting me for the question. We are not working with Brachypodium. We adopted Setaria viridis because it's a C4 model. Brachypodium is a C3 plant regarding the photosynthesis mechanism. And we adopted Setaria especially because of sugarcane. So here in Brazil, sugarcane is a very important crop. So we produce ethanol and we use ethanol in our cars. Myself, particularly, I only use ethanol. So because of sugarcane as an important crop, being a C4 plant, we decided to adopt Setaria viridis as a model because it's increasingly becoming an interesting C4 grass model in the past 10 to 5 years. So no, not Brachypodium. And for sugarcane, a lot of people are working with sugarcane and transforming sugarcane. Sugarcane is a very complex plant because if you're used to Arabidopsis with the diploid plants with the genome less than 200 megabytes, sugarcane, the varieties that we use are dodecaploid with more than 10 giga bases in their genome. So they are very complex. They're massive plants. But now a lot of groups in Brazil are managing to transform sugarcane. And we have a lot of transgenic sugarcane being tested in the field, like resistant to pathogens or with modified fiber characteristics. So even in my lab, we're already working with people from J-Bay in the United States, engineering lignin metabolism in sugarcane for this bioeconomy and for the industry. So in sugarcane, yes, a lot of people are working, including us.
Thank you, Igor. One last question to you, and this one is from Prashant Pawar. He'd like to know whether you found differentially expressed genes in other cell wall biosynthetic processes, cellulose or xylem, for example?
Yeah, yes, we did. We did. For cellulose, it was interesting because cellulose. We found that some cellulose synthase genes that encode cellulose synthase, the enzymes that produce cellulose, they were differentially expressed, but they were not related to secondary wall cellulose synthase. The interesting thing is that for sugarcane, it's not really clear if we do have these differential classes of cellulose synthase. So people from Australia has been working with cellulose synthase in sugarcane as well, and different from Arabidopsis that you have clearly 3 sets of genes for the primary wall and 3 sets of genes of secondary wall. In sugarcane, it's a little bit fuzzy. So our data corroborates their previous data as well, that we see cellulose synthase genes going up, but they are not the orthologs of the secondary wall caesars. And this is very interesting. We still don't know what does that mean. And for hemicellulose, yes, but I have to say that I don't remember exactly which genes are differentially expressed because, yeah, we were really focused on lignin. But I do remember that some IRX10 or something like this for the xylem backbone, I think they were upregulated as well.
Thank you, Igor. With that, we'd like to move on to our last and final panelist of the day, Emmanuel Faria, who is the president and principal researcher for Verriclear Natural Skin Essentials Limited. Verriclear is a company that harnesses data mining to create effective phytomedicinal treatments for neglected skin diseases in marginalized markets and pervasive skincare needs globally. I got to know Manny several years ago when he wrote to me about the work that we had been doing on essential oil profiles, collating and creating, data repositories, public data repositories. And over the years, we've been collaborating, and I've totally loved his quick-witted humor, bordering sometimes on hysterical some days. And in fact, today he addressed one of my serious questions to Benji rather philosophically on the panelists chat box. He thinks others can't read it, but I'm making it public now, Manny. So Manny thinks that compared to dogs, cats are kind of depressive loners, and so it makes more sense that they turn to drugs rather than a thrown tennis ball. Okay, Katie, let's have his slides before this explodes altogether. Thank you.
Hello, my name is Emmanuel Faria, and I'm the founder and formulator at Verriclear Natural Skin Essentials. For the past 2 years or so, I've been collaborating with Dr. Gitanjali Yadav and Peter Murray Rust. Today I have the honor of describing to you our work in progress, the development of an open source toolkit for searching and scraping high quality data from open science sources. The proposed toolkit, which I like to call Dave, is intended to download, abstract, verify, and extract high-quality open-source research data that can be relied upon for effective decision-making and action-taking. Now, the challenge that our toolkit is aiming to overcome is what I like to call the innovation bottleneck. You see, research portals like Europe PMC and PubMed are essentially vast catalogs of millions of research papers. And of course, while it's certainly desirable that the publication of new research continues, this exponentially increasing volume of accumulating research data has already drastically outpaced our ability to make intelligent or effective use of it. It effectively cripples our ability to leverage the existing research to discover and produce new and innovative solutions to the world's age-old and new problems. Dave will download hundreds, thousands, or even millions of research papers, automatically and intelligently analyze the corpus of downloaded papers to identify the relevant data points, scan them using custom dictionaries of categorized keywords, and even aggregate and reinterpret the data that is already contained in tables within each of the articles in a way that can be then output, uploaded, and integrated offline into whatever system or tool that researchers will use to make the decisions related to their desired outcome. So perhaps using the remaining time here, the best way to do this would be to take you right here to our GitHub repository and show you some of the dictionaries that we've put together. I'm going to go in here to dictionary. Inside dictionary, there's an index, and from the dictionary index, there is a description of each of the dictionaries that we have completed so far. One is called EO plant, essential oil plant. This dictionary has 1,678 plant names. They were extracted from the first 186 test articles that we downloaded from PubMed. So that's a lot of names in just those articles. And of those entries, 1,567 had their names normalized separately afterwards and tagged with corresponding Wikidata IDs that will also have descriptions. There's EO plant parts, geolocation. This is the area where the plants were harvested from, where they were grown, where they were processed. And this particular geolocation dictionary will include such data as the country name, the ISO code, the city, latitude, longitude, etc. Now I'll go back out and show you what the dictionaries look like. Compounds. Here is, think of it as a table. Each of these are columns. So the entry name is the name of the compound. The term is also could be exactly the same. Sometimes if there's more than one way to say the same thing, the synonyms, in other words, will have the term be the one that is chosen, and the name will be a synonym. This one has a Wikidata ID, and from that Wikidata ID, anything else about that particular compound can be linked back and pulled out later. We don't have to have it all on our drive or in these dictionaries. Target organism. Target organisms are the microbes that were mentioned in these same articles that mentioned an essential oil or plant name. So we would assume that research was done to see if those oils or compounds would have an effect on these various species. We have species of bacteria, fungus, and all of that takes us back here to what Get Papers downloaded, and here's the PMC ID number for each of those articles. And in this particular directory, as well as inside each of the different article folders or directories, there are now many more files with much more data that Amy has put together for us. And I don't have the time to go into the detail that I would love to right now, but I will show you one of my favorite things, and that is the co-occurrence tables. So for instance, binomial, and a binomial would be a microbe. So can we find a binomial and a compound that goes together, co-occur in these articles? And here we go. So it looks like Staphylococcus and acetate show up the most together out of those 186 articles. Carvacrol would be number 2 for Staphylococcus. Eugenol number 3, camphor number 4. So this is a good indicator that there are experiments being done. I would put my money on inhibit these different microbes. Now that you've seen the dictionaries, I want to show you a little graphical representation of how they will be used or can be used to identify data that might be useful to you. So starting with this case, we have plants. Plants have names. Plants are also comprised of chemical constituents, which also have names. Plants also have parts, one of the dictionaries that we had. Plants have material processing histories, geographic locations. They also have extract types, balsams, absolutes, essential oils. Extract types can exhibit various properties, such as properties that are useful for formulation. Properties can be chemical, density, color, smell. They can be functional. These extract types can also exhibit phytomedicinal activities. They could be antibacterial, antifungal, antioxidant, and also medical or traditional ethnobotanical uses, antacid, anti-Alzheimerian, herbicide. Whew. Well, I wish I had more than 7 minutes to introduce you to Dave and show you the many other different and useful ways that Amy can parse and display data, and to tell you about the many new features that Peter and the team are working on right now to make Amy even smarter and more useful. But if what I've shown you so far has already piqued your curiosity, and you're interested in learning more about Dave and our CEV Open project, we invite you to check out our progress at GitHub on the link below. We look forward to having it ready for you to put to good use in your projects really soon. Again, my name is Emmanuel Faria, and I really hope you enjoyed watching this presentation as much as I enjoyed making it for you. Thanks for watching.
Thank you, Manny. That was really neat, and especially because I've been trying for many years to try and present my work, our work together as well, but I haven't been able to do that. So my first question to you is to get to know you better, for our listeners to get to know you better. So what was the first key moment in your life that turned you onto plants? And what about your current project? What is it about your current project that connects you to that moment?
Okay. First of all, I want to apologize for that little commercial at the end. I wasn't expecting that little window to pop up there on the YouTube. That was embarrassing. And also, hi, Dr. Gita. It's great to be here. And I wanted to say that I'm grateful to the people at PLANTAE.org for hosting this event. It's marvelous. And also to say that I'm really honored to be invited here on this panel today, along with my fellow botanists and scientists actively conducting research. And yeah, I hope I can contribute something meaningful, or at least accidentally, humorous to this conversation today. So turning to your question, I fell into this by accident, like most things in my life. It was 2008. I was the founder of a very large company that was affected by the global credit crisis. And every single day of that following 5 years of that catastrophic market meltdown, I experienced ongoing and also new overwhelming stresses every day that eroded, every aspect of my health and ultimately compromised my immune system. And I ended up with a viral skin condition that, like, many others have no cure. And for me, no functioning treatment. So over the course of 2 years, I subjected myself to increasingly painful and really destructive treatments until one day my doctor just gave up on me and said, they literally told me to go home. And she said, I'll never forget it. Just look, we can't keep doing this because we're just making things worse. And just go home and pray that this, resolves on its own. And, obviously that wasn't something I could allow myself to do. There was just, too much at stake. So I poured myself into PubMed and different clinical study databases to do the best I could on my own. And I felt like, I was using a child's toy shovel on the beach, trying to mine through that literature for any signs of hope. But one of the first things I discovered is that all of the treatments existing for this condition, 100% of them, including cryotherapy, laser surgery, all of them work only 15% of the time. In other words, they fail 85% of the time. And the more I learned about that and began to see different patterns, it was kind of like that movie From a Beautiful Mind, although he was kind of nuts, where, there's all those newspaper clippings on the walls and he sort of sees all the connections like lightning going through them. And partially like that other film, Lorenzo's Oil, the true life drama of a father and mother who battled against the odds to save their son's life from a rare and incurable disease, right? But when all else failed, their persistence led them to a cure, which, saves their boy and rewrites medical history. I'm hoping to do both, but at the time I was just worried about myself. So I formed a hypothesis and set out to prove or disprove it on myself. And I hyper-focused my search at that point, all the queries and parameters to include keywords like plant extracts, botanical compounds, phytomedicine, things like that. And I found that a few doctors had reported some success with just like one thing, just a certain plant oil, or for instance, just pure like vitamin E capsules that they would break open and put on a person's skin, and that worked. And from those few successes, I refined my hypothesis even more and then began searching for plant extracts that had certain constituents and properties and where I could find them, right? The activities. So it was really difficult to find that kind of information, which is how I ended up finding you on S-Oil. So I put together my first formula. It was 15 ingredients in a ratio that made sense to me, and began applying it twice a day. And 110 days later, all the signs of the condition were gone, even down under a dermatoscope or like a magnifying glass, that all of the line prints and the fingerprints had returned as if nothing had ever happened, which was amazing because by the end of all those treatments, it looked like I had leprosy. It was really bad. And so that's when I became a man on a mission. And so far, everybody's had 100% success with this product. And they've even found other uses for it as well, like acne and even under eye circles and things that I wasn't expecting. Well, I guess I could have expected it, but I was not focusing on that. Let's put it that way. So that's how I got here.
Lovely. So with this project now, with drug discovery, the way you're continuing with it, what's your biggest question that you feel driven to answer now, and why?
The biggest one that drives me bananas is not necessarily a question in botany, but in immunology, really, and that's inflammation. What I'm trying to do is, rather than the difference that I found is that all the other treatments were focusing on a symptom and not the root cause. And that makes sense because of the way that some of these companies are what their business model is, right? They don't want to cure you. And I'm not a conspiracy theorist and like that. It just made sense. And so when I look at the situation, I look at it as syndromes, which are combinations of diseases. Diseases all have symptoms, and all those symptoms and the diseases themselves have pathogenesis pathways, and each of those have steps. And I look at it as, a three-legged stool. Why farmers use three-legged stools is because they can be stable on any surface. A four-legged one wouldn't, right? If you go to a bar or restaurant, you know that. And so I look at all of those different layers as if there were a pyramid of three-legged stools, and anywhere I can, I'm looking for a plant oil straight out or a component thereof that'll take out one of those legs on any one of those layers, so that thing will come crashing down, and then the skin can heal itself. But inflammation is a mess. Like, there's just so many pathways to it. And some inflammation points of it are good because they help in wound healing, and others are not. Where you can get into, as we know with COVID, but that's a whole other story. But with allergies and things like that, and so I look at it as a big map. If I had a map of New York City, let's say, and I wanted to block certain streets to keep terrorists from bombing some area, I also want to keep other streets open so that ambulances and fire trucks can get back and forth to the other side to keep people alive and the city going. So if I can find someone to help me figure out which of these things I can safely try to turn off and which ones just don't touch these, got to keep these lights as green all the time, no reds there, that would be a huge win for me.
And I think that's where we should be going with this project altogether. My last question to you is that your talk today, out of the 4 that we saw, was a refreshing advocacy for open science. And it reveals the extent to which full access to literature can truly change, especially your story about your own experience, how full access to literature can truly change the game for a researcher or an individual like yourself, looking to find patterns in large data sets or solutions to questions that you otherwise don't get. So with your experience, do you have any suggestions or recommendations for phytochemistry students starting to work on specialized metabolites? For instance, where could one find the most comprehensive repositories on active compounds in volatile emissions, for example, or anything else that you felt that you learned with your experience, which others starting out may not have at the outset?
Yeah, well, that's a good question. And it did take me a long time to get my way around it and see where to look for what. I didn't even know what searches to make. And the ones that I found kept leading me to, aromatherapy sites and things like that weren't useful at all. But governments have a lot of interest in creating databases for plants, particularly for agriculture, not only for growing the plants or nourishment of the soil, but also pest control. But also bioscientists have done an incredible job of creating genomic information and making this accessible. And each of them offers something that the others don't. So for example, the first one, the most useful one to me in terms of having plants and their constituents and their relative quantities, that took me a long time to find, was SoilDB, yours, Dr. Gita. And I'm so grateful for that and for you, because that's how I met you, of course. And it offers data that was manually extracted, from many text sources, and it includes plant names, the constituent compounds, the relative percentages, the plant material history, including geographical location where they were harvested, stress conditions under which they were grown, some activities as well. So that's very useful. That's at nipgr.ac.in/ESS for essential oil DB. ESSOILDB. Put that in the comments as well. The other one that I found recently, which would have been used to me a long time ago, was Dr. Duke's Phytochemical and Ethnobotanical Database. And it's a combination of some plants, some compounds, some ethnobotanical uses and activities, but it's not really in a usable format. But if you poke around a lot enough, you'll find something of use there. And you can find that at the USDA website. So just look for Dr. Duke's Phytochemical and Ethnobotanical Databases on USDA website. Another interesting one for nutrients and composition would be Food B. It's like Food DB, but no double D. Food B, FOODB.ca. Very interesting one there. And another one is GRIN. GRIN stands for Germoplasm Resources Information Network. And that one is at www.ars-grin.gov. ars-grin.gov. So that's where I'd find information. And the other kind of useful tip or trick I'd like to put out to anybody, because I've been sitting in this chair for a long time, staring at walls of data and spreadsheets, and it's really tedious work. Cleaning up these databases and just finding out the dictionaries rather, and finding out which words are important. What do I want in there, and how do I separate them and classify them? And so, for someone who is little ADD, you might notice that, like me, it's difficult to concentrate. But I do two things. One is if you search for brain beats or binaural beats, you'll find some apps for that, which are really great. If you want, figure out how to write to me, and I'll tell you which ones I use. But basically, you know how dogs can only hear, dogs can hear certain frequencies that humans can't. Well, the way brain beats works is to get you into, for instance, an alpha state, brainwave state, or a beta state where you really want to be hyper-focused, or a theta state where you want to chill and relax and have ideas come to you. It'll play one frequency in one ear, a different one in the other ear, and then if you remember basic science about waves and troughs, it'll trick your brain into hearing and responding to, and then being entrained in that frequency. And for me, for focus and concentration, that has been, everything. And then the other thing is I have these hue light bulbs that I can change the color on, and I've been playing with certain frequencies, I guess, of light as well. But orange if I kind of want to like perk myself up, green for concentrating, red for really relaxing, and it's actually very good for renewing the cones and rods of your eyes. This is new research. So those are my two big things that I hope will help everybody to be able to stand, no matter how interested you are, but stand doing that tedious part that gets you to those dopamine hits every once in a while when you actually get to the destination.
All right. So in the meantime, what we're going to be doing is, while Manny goes and comes back, I think I would like to take a couple of questions for the other panelists. Can I request all the panelists to please switch on your videos, because I'll be asking questions to each one of you from the audience, essentially, that we haven't done earlier. And the first question is to Greg. Greg, this question has been put to you by one of our listeners, Samuel Smith, and he's asking you about whether you're planning to expand the metabolite marker profiling to other isoprenoids, for example, sesquiterpenes, monoterpenes, and so on. Would you like to take that, Greg?
Oh, in fact, no. We don't plan to do that, but maybe that our method can have the ability to identify and quantify more than 60 compounds in one single run. But we are limited by the extraction. I think we use methanol, and maybe not the best solvent to extract monoterpene, for example.
Okay, you can take your time while I talk to Benji about a question that has been put to him by Brenda. Benji, the question to you is, it's again a cat question, and I know you must be tired of listening to more cat questions, but is it known how many species of cats respond to catnip?
Yeah, I think it's kind of the Felidae clade, which includes like panthers and tigers, yes. Leopards, and I think many cats. I think they've obviously, dogs aren't responsive. I don't think bears are responsive, but lots of species.
You had mentioned this in your talk, but I still just ask the question again. Thank you, Benji. Manny, would you be willing to take the question that I typed out for you in the chat box? The question essentially was, about open science and about advocacy for using large-scale data-driven discovery for biology. So would you, the question to you again is, in large data sets, finding patterns that are useful for biology? To what extent do you think we need manual intervention to remove false positives? Especially noting in your talk that you had ranked, and in one of your co-occurrence tables, you had this improbable correlation between staph and acetate. You did mention a couple of things about it, but I would love to know the extent to which we can, rely on automated methods, and what kind of manual intervention do we need? Thank you.
Oh yeah, the mistakes. Well, there's actually two mistakes in that presentation I noticed. One was that I noticed that it said there was 1,600 plant names that came out of that first 136 test articles, but actually a majority of those plant names came from SSoilDB, Dr. Gita's database. So that was like the first seeds in our work there. Oh yes. So the thing about the acetate, right? That came up? No? Yes? Okay. Sorry, working deaf here. There's a number of things. First of all, that slipped my attention, but what's interesting is because it's open notebook study, it's actually how we work, where we make a mistake in real time and we learn from it and discuss it among friends and put it out there. We don't wait for 2 years to write something and then publish it. So you got to see that first thing. But first of all, because it was so late at night when I put that thing together, I picked the wrong folder, and that was actually our very first set of tests that we did. It was an old dictionary that wasn't normalized yet, first of all. Secondly, it was an old version of the software that picked up individual words instead of words that are together. So there are obviously various kinds of acetates that were being mentioned in the literature, but it was picking it up as a single word and putting that at the top because it came up so often, obviously. And I forget that last bit. But
no, don't worry about it. Don't worry about it. I'm going to be very short on time. We're running out entirely. So I would like to put my last question to Igor. And this question is from Benji. So Igor, Benjamin is asking, network analysis gives you more information than just using clustering?
Well, they basically provide you the same kind of information, how similarly expressed the genes are. Clustering actually provide you more information because it shows you in which tissues, let's say, or in which samples, how much a gene is expressed. Let's say that you have like a heat map and you cluster those genes and you have all the tissues in one axis. And the genes here in the y-axis, and then you can see the expression values more obvious that the network that just provides us whether two genes are more or less co-expressed. So you have advantages and disadvantages of both. I think in a way it's easier to represent a network with a lot of genes that you see the subclusters of genes which are interesting. They are clustered together with strong connections, and then you have other neighborhoods. But the clustering also can show you additional data with the expression values or how similar two genes are expressed. So you have advantages and disadvantages. You also can represent co-expression analysis as a list. So you can just have a list of genes that are co-expressed with your single. Of course, you have to select one gene, and you want to know which are the genes that are co-expressed with your target gene. And then you have a list of genes, and they are ranked based on the expression coefficient. So then you can have a list and see which ones are your interesting targets within this list. So basically you have lists, clustering, and networks that have advantages and disadvantages.
Thank you, Igor. And I'm sure Benji and you can get in touch with each other if there's more that you need to discuss. I would like to close now. Hold on. There is this comment by Amanda Storm. Well, she agrees with your cat hypothesis. I wouldn't have believed anybody would say that, but she does. So, all right, great on philosophy.
College knowledge, yeah.
The question she asks you is there anywhere that walks through or explains the process of using Amy? And I'm dying to answer that question, but you do it. You take it.
Oh, okay. The best place to look is to go on GitHub. And the link is a long one, and I have it here somewhere.
Go for
GitHub.com.
Okay.
Yes. Yeah. GitHub.com actually /PeterMR github.com/openvirus/wiki/getpapers. Just a short one, but you know what? I'll paste it into the chat, and hopefully it'll show up for everybody.
And I'm going to request Katie to put it onto this answer box in the main Q&A. But essentially, Amanda, this work is being done live as we speak with Dr. Peter Murray-Rust, who is at the University of Cambridge, and we've been collaborating for the last several years. This is fantastic amount of real open world science that we've been doing together in extracting knowledge from literature resources, open source literature. And you're welcome to take a look at Peter Murray-Rust's GitHub repositories. All of them will give you enough information about Amy, but you're more than welcome to check out CEV Open. We call it CEV Open because it's about content mine. That's Peter's essential oil database, which is mine, and V for Verriclear, which is Manny's. So CEV Open is what works for us. Thank you very much. With that, seeing that there are no more questions, may I request Katie to take over? And first of all, thank you all of the panelists for your talks, and thank you all listeners for staying around with us here. I especially invite you to join us on part 2 of the PLANTAE panel on specialized metabolites on February 3, same time next week, same channel. Katie, over to you.
Certainly. Thank you all of our panelists for joining. Also, thank you for submitting videos. This panel was selected from, we had really great submissions. And so that's why there's this part 1 and then part 2. We're really looking forward to the next one next week. I set in the chat a link to where to register. Thank you all for joining, and we'll see you next time.